hello, hello. Hello, everybody. We have Dr. Dan, we have Scott, and we have a rational empiricist. And so, if you imagine like the problems in the world for humans, like the nuclear weapons problem, and you can imagine the hurricane problem, right? And if like the nuclear weapons problem and the hurricane problem got together in the same place, that would be what happened with Rebekah and Cell getting back together on her channel. so that's a video we're going to be reviewing. So anyway, Dr. Dan.
I'm sorry. Do you think Sal became less homophobic, or Rebekah became more homophobic in order for this to partner back up?
In order for the union, right? Maybe they decided that, oh, gee, we don't have to agree on everything like normal adults, right? I don't know, but could have happened. Hard to say. So, and there's a couple things in here where Cell talks about Brandolini's law that are significant. It's all the way through, where he talks about, oh, the evolutionists, they just don't think about this stuff, and so he imagines that nobody in evolutionary biology is thinking about the problems of protein families and gene gene creation, origination, any of that stuff. And he talks about Brandolini's law. And then you did a video, Dan, on Casey Liskin, where he was complaining. He was saying that we don't, we hide the evidence. And then you showed, and I linked it in the chat and in my description, you showed that he was doing that. And it's kind of like if you looked out and somebody was racing around your lawn in a Dodge Charger, ripping up your lawn, and you went out there to yell at them, and they yelled at you and said to you, "Get off my lawn." Yeah. I mean, it gives you that same feeling of frustration.
I know.
It's like, that's what you do.
And it wasn't like, the great thing about the Liskin thing is that's almost like identical to the thing Michael Behe did like 15 years ago with this table with polar bear mutations, where he said all these mutations that we've documented are harmful. And the table that he used, he actually cut out several columns and a bunch of rows from the table, and Nathan Lentz actually went back through and found the original table and showed like, hey, you left out a whole bunch of data, bud. And then Casey doing basically the exact same thing here. So
right, and then Tompkins with that, where he took that picture. I didn't hear you, Andrew.
It's amazing how 90% of what comes out of their mouths is mostly projection.
Yeah, I know. Yeah, and the other thing, Tompkins, where he took that picture of the banding between him, human chimps and chromosome 2, or chromosome 2 and chimps, and somehow he stretched it to make it look like it was different, and it wasn't. If you look at the original, I can't see a difference. They just line up perfectly, but yet he did a little cartoon drawing, and he put it in the same place, and he referenced the paper, and somehow he stretched it. And it's just like, come on, guys. Maybe they do it, maybe it's like they're not doing it consciously. They're just doing it subconsciously. I don't know.
I have no idea.
No idea. Yeah. So,
higher purpose, higher cause in their minds.
Higher purpose, right. I think I'm going to share a screen. So what do you think?
That's my best guess.
Yeah, I think, it's weird. It really concerns me. It always concerns me that I'm doing the same thing, right? So I'm always trying to think, am I doing this? Am I being biased? Am I, missing something? And of course, they would absolutely be sure that I am. But when you run into two people that are looking at the same piece of evidence and coming away with two different things, it makes you wonder about whether human brains work at all. I don't know. Let's see. I'm going to present a screen. I'm going to try. I'm going to try to do this right tonight. Got evidence for evolution. Share. So she's on episode 9 or 10, I think, of this evidence for evolution.
It was at least one episode after this one, which
there is.
I watched this one. I did not sit through the most recent one.
Seems like she did a video on this channel compared to her older one.
Yeah. She did a video basically as a comeback to this video, to her own video, right? Because Grayson was on there and he mentioned a bunch of stuff. So she's done another one. So we'll just see what we got here. I think this is kind of the beginning.
By sharing some evidence against evolution. And if anyone joins, then we'll go from there. So I was shocked yesterday to find this paper, as this is in a peer-reviewed journal, and I was absolutely shocked by the title. It says, "Biological evolution is dead in the water of Darwin's warm little pond."
Okay, so she's shocked to see it in a peer-reviewed journal, and she's sort of implying that that was a, you know, that would persuade her. Yet she's not persuaded by the peer review, by the hundreds of thousands or millions of articles on evolution in peer-reviewed journals that, you know, find for evolution. But now this journal, Progress in Biophysics and Molecular Biology, has Dennis Noble as one of the editors. Guess what? And these two guys, Brown and Hollander, a mathematician and I think some kind of biologist, I'm not sure. They've done a few of these papers basically where they come out, and they did the usual probabilistic bullshit, and they did it with enzymes and let's say the improbability of enzymes and vitamins co-evolving, that sort of thing. So
yeah,
it kind of showing that life is impossible, you know, that kind of thing.
For anybody who wants the full Dennis Noble treatment, you should head over to Zach Hancock's channel at Talk Pop Gen, and he's got a whole video on Dennis Noble and all his nonsense. And if you go to this paper, you'll see kind of beat by beat that playbook. It's not good. It's not good evolutionary biology. It, in fact, ignores a lot of the principles of evolutionary biology. Yeah, it's not great.
Hey, as a quick parenthesis here, didn't you announce with Dr. Zach Hancock that y'all were doing a paper that's upcoming, or?
So we had the one last year on genetic entropy, and there's something going on with the 100th anniversary of the Scopes trial, and we'll see what comes of it.
Okay. All right. Yeah. Okay. Stay tuned.
Just post it a little.
Stay tuned. We'll see. I don't know if it'll happen. We'll see.
Yep. Oh, let's see. Hollander. So I researched Hollander and Brown a little bit, and they do biological evolution didn't happen. They've done a few videos and stuff like that. And these guys, not a surprise. And this biophysics journal has put a few of these articles in here. And if you go and look at what is his name? Sandwalk, the guy on Sandwalk.
Larry Moran, yeah.
Yes, he did a little thing on this, and all he mentioned is that Dennis Noble's the editor, and he felt he didn't need to say any more. Dennis. Yeah, and they're writing opinion pieces. So,
why do people keep doing this? Why do they publish in a biophysics journal and not in ecology or
right? Yeah,
because the peer reviewers in an evolution journal would know that it's nonsense.
Yes. Right. Or biophysics, they don't. Okay. So let's see. I think I'm at a different timestamp. Let's see what she says here.
Specific problem that's being referenced in this paper, and why they say that biological evolution is dead in the water of Darwin's warm little pond, is because they are picking out one specific problem, which is the co-origination of enzymes and vitamins. And they're talking about how, look, natural selection really it doesn't have the power to facilitate things that have dependencies on each other in order for origination. Meaning, if you have an enzyme, which if you're unfamiliar with that term, it's really a machine.
Right. Okay.
Right.
Okay. Anyway.
So this is an irreducible complexity argument, and the
Right.
underlying premise, in addition to the just being wrong because irreducible complexity is wrong, the underlying premise here is that the first forms of life would have worked the same way as currently existing forms of life. Nobody thinks that.
Right. Yep.
Modern
and if you look at
the first life forms.
Yeah. And if you look at the enzyme structure, the structure of those particular molecules, and you look at at NADH and stuff like that, and you start reading about the early evolution of the citric acid cycle and how all of those chemicals they're connected in so many different ways synthetically. It's really amazing to watch it.
And you can have abiotic citrate-based redox reactions.
Yeah.
So you don't even need cells and enzymes to do it. It just happens spontaneously under the right conditions. And we can find those conditions on Earth.
Can I ask a non-scientist, I can spell science, and that's about as close to being a scientist I am. Enzymes are just
no.
No, okay, sorry.
No, you can't. Go ahead, Scott.
Enzymes are just chemicals that are helpers, right?
They're proteins that accelerate chemical reactions.
Okay, so helpers, and it helps two reactions take place. So instead of thinking binary, that there has to be two chemicals present in this process of natural selection, there has to be three. One of which it helps facilitate the reaction. I don't see why anybody has a problem with that.
Well, there are some that need additional support for the enzyme, like vitamins and stuff. And I think that's what they're doing. Unfortunately, this paper I cannot get it. Dr. Dan, if you can get this paper for me, maybe
probably get it. But also, it's like we don't need to spend time on this paper because it's just irreducible complexity, and that's wrong. And in terms of you need vitamins. No, you don't. A lot of enzymes just use metal ion cofactors instead of organic cofactors. And wouldn't you know it, where you're going to find metal ions that could operate as enzyme cofactors at deep sea hydrothermal vents where this stuff
in the ocean. Right. Okay. So now you said you had some bookmarks too, Dan. I don't know how you want to do this. I've got like 40 some bookmarks, so I mean I can go through it in order.
So mine starts at 17-ish minutes because that's where she gets into the nested hierarchy stuff.
Nice.
And specifically where she talks about how they're supposed to all match and then they don't match, and then evolutionists invent words like convergent evolution. And it's frustrating to listen to because that's not how any of this works. So if you want to hit
I've got
17 and we'll go or 15, whatever.
Well, I got a bookmark at 1443, so we'll start there and maybe run ahead a little bit. Okay.
Can be true while the other is false. That means that they're separate. Yeah, but let's not be ridiculous here. I'm talking about, yes, absolutely. But we could separate every point of evolution. We could say, well, not only did God do a miracle at abiogen, at the first living cell, but then he did a miracle halfway through evolution. And he did, and evolution, I think it does need miracles, honestly. So I think that's the only way that evolution is happening is with a whole lot of miracles. But to say that means they're separate, I don't think that's true, because what we're talking about is the idea, the philosophy, the understanding, the worldview that says we came about by natural processes. And that is the worldview that I think actually contradicts our scientific observations of the world. When we observe the things that are around us, plants, animals, ourselves, consciousness, everything, these things point to an intelligent designer. We know.
Yeah, that's the intuition of the common man, basically. That, oh my God, this stuff requires miracles. It's a miracle, that sort of thing. Let's see. You said 17. Do you have an exact bookmark, Dan?
I just in my notes it literally says 17-ish.
Oh, okay. Here's. Oh my God. I was telling. My sons always tell me to show up at 5-ish, and I say I don't do ish.
Yeah,
I'll show up at 5 sharp. Right.
We'll hear it.
I'm at 16:30, so we'll play.
That's fine.
There.
Let's let that.
That is where our
scientific
observations lead us, if we are not trapped into a framework that forces us into thinking according to natural processes only. Right. So, Raised by Wolf says, I mostly accept evolution, but I don't have my day ruined because someone somewhere doesn't immediately accept every single aspect of the theory. Cool. Thank you. I appreciate that.
So what she just said, basically, that if you're trapped into this naturalistic framework, well, methodological naturalism and how science works, basically you can't do science if you have miracles at every point, right? I mean, so we're not trapped by it. It's something that it's how you figure stuff out in the world. You have to look for natural causes.
And it's a requirement because we can't find supernatural causes. Literally, we can't study them by definition. Yeah. They're outside of
they're random
measurement.
Yeah, they're always mad at us
because to figure out how the world works. Sorry, go ahead.
Yeah. They're always talking about us because we use the word random mutations, and it really, I saw a video today where they're really irritated by that. And it's like supernatural stuff is necessarily random. I mean, it can happen anywhere at any time for any reason, right? So we can't do anything with that. Anyway, insinuating, embracing, taking seriously what scientific theories tell us about the world constitutes a worldview. Anyway, okay. So we're getting close to 17-ish.
Well, we're there. Philosophical
1 second.
With methodological naturalism as well.
Yeah.
The same thing.
Yep. Okay.
Evidence and reason. Sal says, even Dr. Dan admits there's no universal common ancestor for all major protein families. Yes, that's true.
You admit it.
There we go.
You admit it.
I admit it that new proteins evolve.
Yeah.
You caught me.
Well, right.
It's almost like we observe that happening.
He's got to bring up the protein orchard.
Yeah,
he's got to bring up.
He's going to show up.
Okay, he's got to bring up the protein orchards, and the interesting thing about this video is he actually explains what he is thinking when he thinks that protein orchards are a problem for evolution. He's talking about the origination of the protein families. Okay, he says, "Oh, that can't happen." So he's reaching into the abiogenesis part of where all of these little tiny fragments and protein domains and motifs popped up. And he doesn't see how that could possibly happen. So he kind of accepts protein families evolving, but then a little later on, we'll see that when he talks about it, he never pays homage to the fact that there are protein families. For instance, he will talk about zinc finger ZNF 136. ZNF 136 is one of 800 zinc finger proteins in the human genome. There are 5,000 zinc finger domains in the human genome spread across 800 different genes. And he never talks about that. So anyway, okay. You wanted to go a little further here?
Let's just let it play.
You know,
okay.
Sal, you never get tired of repeating that, do you? Sal says, common design can also make nested hierarchies. Common design is a better explanation for supposed co-evolving nested hierarchies, such as binding interactions and nuclear localization transport. Sal, why don't you come on and share about that? So I'd like a bit better explanation on the nuclear localization transport. Grayson says, common design cannot sustain nested hierarchies. Mutations would break them. Now, okay, look, nested hierarchies. We've been talking about that a lot, and I'm going to separate this from my Dr. Dan talks because I want to complete that talk with him separately. But I will tell you, there are so many problems with nested hierarchies. Oh, where do I begin? First of all, we have to decide how we're going to group animals together. Are we going to group them by morphology? Or in recent times, we're trying to group them together by genetics. But what happens when we do this? We have problems because we have some organisms and the proteins is also a problem. If you try to organize them by which proteins they have, that will give you a different tree. So if we all decide,
that's kind of like overstating what happens. There are some, okay, we've come up with these genealogies based on morphology, historically, right?
Yeah.
And then we got the genes and the genomes, and we started looking at it. And now they're down to fighting about really, really specific areas about, because sometimes these protein trees will be just slightly different because proteins kind of evolve at different rates. There's all sorts of stuff that happens that makes it a little unclear, right?
And the gene trees can be different too, where you do a phylogenetic tree using one region of the genome or one specific gene, you get one branching pattern. Use a different gene or a different region, you get a different pattern. That's a thing that happens, and it's well understood how and why that happens. And she's going to name in a second one of the mechanisms and laugh about it as though it's not real. Here, play again, and you'll see.
Yeah, she says that we make up words, basically. Yeah, that's what that's not quite what it's called. We don't just make them up just for the hell of it.
Descended from a common ancestor, then you would expect that everything would match up, right? Morphology would match with the genetics. It would match with the types of proteins that are in each organism, but that's not what we see. And what I've noticed is that the trees, when they get broken, basically when the tree doesn't work, evolutionists invent words to try to make it okay. Oh, it's all okay. We have convergent evolution. So convergent evolution is just a word that's made up to patch up a major problem with the tree of life. And that
there's this thing, when we see something that doesn't seem to make sense, we have to go out and try to figure out, well, what could have happened here, right? And so then we figure out the mechanisms, duplications, horizontal transfer, all of these different things, convergent evolution. The words aren't just made up. We get them from the mathematics of how genes mutate and recombine. And then we go out and stuff. These words are not made up. They're individual hypotheses, basically.
I think there are 2 points that can also be made here, and I'm sure Dan was probably also going to say this. For one, what she was saying earlier, that this certain amount of phylogenetic incongruence is completely unexpected. No, it isn't, because, for example, for certain population genetics processes related to incomplete lineage sorting, we can actually predict the specific amount that we should observe, especially for recently branching lineages or rapidly speciating. And second, when she was talking about convergent evolution there, and most of the examples that I'm familiar, at least, it's typically the standard example of the wings of an insect, a bat, a bird. You can pretty easily tell them apart, but you can tell they all serve the same function. Obviously, it gets a bit more complicated when you look at molecular examples. But it's not completely irreconcilable here.
And just to put a point on that, it's not just that we can predict that incomplete lineage sorting happens and we can document it. We can predict the exact frequency of incomplete lineage sorting accurately. So we can predict mathematically how much of the chimp genome should be more similar to gorillas than it is to humans, right? How much of the human genome should be more similar to gorillas than it is to chimps, based on when we think those entities diverge from each other and the effective population sizes, whatever, and you can calculate how much incomplete lineage sorting you expect to happen, right? And we can go back and look and show that those predictions are accurate. And then the other thing with the convergence, a lot of times there's an underlying molecular aspect to it. So it's actually a more specific thing called parallel evolution, where it's actually the same mutations. And you see this a lot with like antibiotic resistance, where you can have the same species of bacteria and independently it will evolve resistance, and the mutations that are selected for in those different patients where it's being exposed to the antibiotic, it's the same mutations that get selected for. So you end up with convergent evolution, not because they're being like directed to do the same thing, but because mutations are just happening. And in both environments, you have the same selective pressure. So the same sets of mutations have the optimal fitness. Again, we can experimentally document that. So there's nothing like made up or ad hoc about any of these explanations. When we see weirdness in phylogenetic trees, by and large, we're pretty darn good at explaining it. And we even account for this stuff. So like one thing when you're actually doing phylogenetics for real, which is something I've done, one thing you have to do is you have to account for recombination, because that messes with your phylogenetic structure. You have to remove regions, and my experience with this is specifically with viral genomes. You have to remove the regions from your alignment that show signs of recombination, because obviously that messes with your ability to show the vertical ancestry of each lineage. So this isn't just like made up ad hoc stuff. We can actually document the mechanisms, predict the frequencies, and show that they're accurate.
You can add more genes to the analysis too, and you can tell that apart as well. Typically hundreds or
just do more.
Right. Yeah.
And coalescence theory is really deep, and the results that it comes up with are very non-intuitive. You end up with things like the mitochondrial Adam and Eve. And that is completely misunderstood, because if you pick any other gene, you're going to end up with a different individual that's going to be basically the Adam and Eve of that, that particular mutation, right? And coalescence theory is non-intuitive. I think.
I agree. You have to really dig into it in order to understand it. Yeah.
Right. Comes up with stuff that
Deep Murder Man has a question here on the screen. I put it on the screen. Creationists say same creator for why things look similar. What would we expect to see, or is it possible to know from a creator? Didn't you do a video similar to that?
This is
breaks down.
It all when you look at molecular data for that, like it's common design instead of common descent, it totally breaks down because mutations have to happen, and unless God is like directly deciding which mutations happen in each lineage, the mutations are probabilistic, so they're going to break whatever pattern was built in at the start.
Right. Yep.
I also know what we see with known human designs, they don't typically fall into a nested hierarchy to begin with. So that explanation doesn't work.
Yeah. I hope Rebekah showed up to watch this. I'm not sure if she has.
Let's see.
So where do we want to go? Let's see. So I've got a bookmark at, Sal comes in at 2059. So we're
so why don't you go to Sal, because he's going to talk about Joe DeWeese and how we don't try to explain anything.
Yeah, I know. Yeah, which really,
By
which
the way,
is the thing.
Real quick question: does Rebekah ever talk about the explanations about the mechanisms of convergent evolution that science has discovered, or does she just say it's a made-up word? Don't believe it.
She says it's a made-up word. She didn't want to hear about it.
So she shows no awareness of what science says about the subject.
Right. And if you try to explain something to her, she'll say, well, that's just a story. Well, a hypothesis technically is just a story, right? Some stories are true stories, right? And a hypothesis is a potential story that could be a true story, and then we test it to find out if it is a true story. So, I mean, So she dismisses you when you try to get down into the details, and yet they'll keep accusing us of never getting into the details, but then she won't listen. And her and I had a falling out over, I think, the evolution of the liver, where she said, "Oh, that's just a story." It's like, I'm trying to tell you what they think happened, right? You ask, how could the liver evolve? Here's how it could have evolved. Yeah. It's like, of course I got to tell you a story. Science communicators, the only way they can teach is by telling stories. You could consider it a story.
I do. I structure my lectures, my classes, they're structured like a story because I have to keep 450 people engaged for 80 minutes. I have to keep everybody awake, right? So there's, even if it's something, talking about the movement, electron transport during photosynthesis, right? It has a narrative structure to it to keep people engaged. Doesn't make it not accurate.
And when my mom drugged me to Bible Sunday school when I was 6 years old, they told us they were going to tell us Bible stories. So, they're just stories too, right? They told me they were stories.
It was trying to level the playing field, basically.
Yeah, it is basically. Yeah. Hang on a second, Thor. You're in my way. You're kind of fat. I got a cat.
So we got Sal coming in here, and then there's a whole bunch of a lot of my notes on this are super repetitive, like Znester hierarchies, but common design. But there's a thing at 30 minutes that we should watch because Sal has kind of a different explanation. And I think it's worth just commenting on.
I have 5 clips I want to play in succession at around 29:41. So 30-ish. I'm pretty sure it's the same thing that you're talking about. So he has a really good explanation for the biology. Right. Yeah. Okay. What was his introduction?
Question about nuclear localization and transport. It's like when you change a key.
Okay, nuclear localization is when a protein is translated, created outside of the nucleus, and it's transported back into the nucleus. And there's something called a NLS, or a nuclear. You can't say this 10 times. Nuclear localization signal. Say that 10 times real fast, everybody. Anyway, so it's a particular signal, and Cell keeps insisting that it's a lock and key. But he does say that it's between four amino acids and 30. And there's a whole lot more to this whole subject. And he accuses us of not digging in deeply. Well, he does not dig in deeply. So anyway, we'll listen to this.
Like, let's say you have a master key that opens all the locks in the building. If you change that key, you're going to have to change all the locks as well.
And what if you have a skeleton key, right? That fits in more than one lock.
Right.
So when you have like a protein that interacts with like 100 other proteins, like say in a plant, and then you do the same for a mammal, what the evolutionists don't do is they don't account for the fact that if you change something that interacts with 100 other pieces, like a master key lock, versus all the locks, you're going to have to change all 100 simultaneously. Especially if it causes death. That's pretty bad. And you don't.
Okay, right there, he hits on it, right? If it causes death, it ain't going to end up in the genome, right?
Right. Why would that be if it mutates in such a way that it causes death, then you can ignore it because it's never going to persist.
Correct. Yeah.
What's the I don't understand what the problem that he's trying to articulate is.
He answers his own question.
One pedantic question, real quick. When we talk in terms of a mutation that causes death, we're talking about that causes death before reproduction is possible.
I interpret it as insta-death, basically.
Okay.
Yeah. A lot of that stuff. Yeah, a lot of that stuff would cause death before there was even a pregnancy. Basically, the embryo wouldn't even embed.
Right.
So, it wouldn't even happen. And like God is the greatest aborter of all times, 30 to 50% of all fertilizations end up on the cutting room floor, because of mutations, that sort of thing. I'm trying to find something here.
Is he talking about protein interactions specifically, or all molecular interactions, because if you look at evolutionary development, that's clearly not the case when you're talking about like transcription factors affecting multiple genes. I don't know if he's thinking about that specifically, or
I don't know if he knows. I think you're grafting more specificity onto his analogy than is there in the first place.
okay.
Yeah,
Fair enough.
he's got a bee in his bonnet about nuclear localization signals, which he
Okay, so he's
found in
focusing on
his
that, yeah.
topoisomerase gene, right?
Right.
So there's like in the C-terminal end of the protein, way on the end, there's a little tiny address that basically causes it to connect to an important. It's a protein, and it helps it to go back into the nucleus, right? So it's like an address. But the thing is, this address is called, it's a sequence logo. It's a sequence logo is not a specific address like you would put on your envelope if you want your envelope to end up at somebody's house. It's an address that you would put on your envelope if you want your envelope to end up in a county, or maybe a city, or in an area, because a lot of the addresses are going to basically go to the same spot. They're going to do the same thing, and he leaves all that out.
Nucleus is a big place.
Yeah, and I have some diagrams where I can show you these which are kind of cool sequence logos, basically. I'm not sure if I can pop one up here. Let me show you one. Oh, let's see here. I'm going to cut it out. Okay, I think this is worth showing. If you bear with me here for just a second. Well, my Mac pops it up, and there it is.
Yeah, the point of all this, everybody, is that biochemistry is messy and squishy. So one sequence will usually stick to more than one target, and lots of sequences will stick to the same target.
A lot more flexibility than they give it credit for.
It's a lot more flexible. Yeah.
Yeah, here's a sequence logo. Okay. Let's see. This is a DNA sequence logo. And what it's showing you here is...
Wait, we're still looking at Rebekah and Sal.
Oh, God. Okay. I'm sorry.
I don't say anything.
Why? Oh, my goodness. Oh, no. Let me go to settings and see if I can figure out.
You should be able to take that screen down and put a different one up. You should just...
yeah, I don't want to do that. I've got OBS here.
Yeah, all right. Don't worry about
let's see. Oh, there's OBS.
Yeah, Nestling's on it. Yeah, folks, what you should understand is that there's just a lot more flexibility than Sal is letting on in how these interactions work.
Right. Well, let's see. Okay,
even his key analogy is faulty, because you can have keys that work through multiple locks through like different means. I just looked it up just to see if he was wrong, and of course Sal was wrong about his key analogy.
Yeah, and you've got promiscuous proteins that have multiple targets, so it's like a key, but it's got two other keys sticking off of it, and each one binds to something slightly different. Right? There's biochemistry is messy. Yeah. Nothing works. Nothing works the way he's describing. We should jump. Go ahead.
Yeah. Okay. OBS just did an update, and it makes it impossible for me to do anything other than take this down. And but anyway, I wanted to show you a sequence logo because maybe we'll get to it later. Because I think it's important to understand to respond to some of these criticisms a little more technically than we do normally. Okay, so we're let's see, where are we going here? So
let's
we're
do
at NLS
29-ish thing.
Okay. Oh, you want to do that?
Let's because it's such a perfect insight into the response to nested hierarchies.
Yeah, it's going to take. This could be cleaner, but I'll do it the best I can here. So 2924 is the first one.
Yeah, that's fine.
And I think it's we'll do it.
I like the way Ration Extrema puts it while you're looking for that.
What's that?
That's a good way of describing all these biochemical interactions.
Yeah, exactly. Just bumping into each other until they stick together. Yeah.
It's like nothing mystical about it. It's just
the creator can do.
Here we go. This is it.
Okay. Let me go do that again. I'm sorry, Andrew. I didn't mean to start it while you were talking.
Oh, no worries.
Okay, here we go.
I mean, a creator can do anything. So, sorry.
But okay, Dan.
Okay, and then let's see. So
yeah,
2924.
Grayson does a good job restating it. So let's let it. Yeah.
Yeah,
Sal's
and then
gonna
20. Oh, you want me to play that part?
Yeah, just let it go for like a minute or two, because Sal's going to dig himself a deep hole here.
Okay.
Dan's argument was that even if the creator created a perfect nested hierarchy at the start, it's the accumulation of mutations post-flood, post-fall, that would break that nested hierarchy.
A creator can do anything.
Okay,
you agree though that these mutations, okay, if you're accumulating random mutations, you would not expect these mutations to be in the same places if they're accumulating independently along separate lineages that we're not sharing a common ancestor.
We don't know the mutation rates. This is
what?
To extrapolate what we observe now and then extrapolate in the past. And fundamentally, a creator can do anything. He can accelerate. A creator can do anything. He can accelerate. So I'm sorry.
No matter what mutation.
Can you mute your mic when you're not the one talking? Because there's a lot of feedback coming. So.
Oh,
but,
really?
Yeah, it's very noisy
Sal?
on my end.
Okay, Sal says a creator can do anything about 6 times in this video. He just keeps doing it.
So he says that the creator could be intervening as things are diversifying post-flood, right? The creator could be intervening to maintain the sequences in a nested hierarchy even as mutations are happening. Okay,
and why would he do that?
We're not in science anymore. We're completely just ongoing miracles. Great, cool. You can believe that.
Position.
There is a passage of scripture that many, many, many preachers and theologians love to quote, which just says, God is not the author of confusion. And that's what you would have if God was just making mutations pop up everywhere, doing anything and everything he wants to do. It basically sounds like he's my 4-year-old grandson when he wakes up, has his breakfast, and then suddenly he's just Dr. Chaos all over the house, throwing things out of cabinets and refusing to put them back in. When I come to take it away from him, he runs away and throws something else. You know, that's the God that Sal is describing, an author of confusion and disorder. And so Sal right there just admitted he's not into science. He's not a scientist, because I say we like I'm a scientist. Science is about finding the order in the chaos of information, not really the chaos of actual reality. But of course there is chaos in reality, but I'm going to stop talking now because I'm going to ramble. But yeah, it's just this is a striking lack of consistency and even theology on the part of Sal. He's gone off the deep end.
A creator can do anything, and we have to explain all the details, right? We can't do it that way. We can't dismiss it like that.
Mm-hmm. To be fair to Sal, don't do that. Every creationist is as bad as this. Nathaniel Jeanson, Answers in Genesis, Harvard PhD, in one of his blog posts in the Answers Research Journal, one of his popular level science articles in the fake journal Answers in Genesis, he talks about how God upholds his creation via means. Those are his words and Jeanson's words in this article, and how he actively works to uphold his creation in the present. So Jeanson is describing the same thing that Sal is describing here, where God is actively intervening to make things be a certain way, which is a completely ad hoc, untestable, unfalsifiable, non-scientific position to take, right? So the to be fair to Sal here is to say that he is not out on a limb making this terrible argument. He is in company with other professional young Earth creationists who certainly should know better making this terrible argument.
I mean, I'll note that, like, even Newton, for example, he kind of sort of made the argument God must be upholding the celestial motion of the solar system. But then what did we discover later on? That we didn't really need miracles to explain the motion of the planets. So it's when your hypothesis can accommodate anything, it doesn't really explain anything.
Yeah.
And this is why theology is not science.
Yep.
If you have a non-conserved sequence and you look at the chimp and you look at the human, and there's an ALU right in the middle of that intron, right? That's identical between the chimp and the human, and the ALU does nothing. That's a transposon, or Donnie's ERVs are a version of transposons. Anyway, so if you have an intron basically between a chimp and a human, then you go look at an ape, and it's not, or a gorilla, I'm sorry, and it's not there. And it's like, okay, why is it just in the chimp and the human if we're separately created? Because it doesn't do anything. Yeah.
How do you know? How do you know it doesn't do anything?
Yeah.
We found tons of uses.
Yeah, right. We don't know. Yeah.
Right. We don't.
Yeah, like
look, that's just an appeal to ignorance, Mike. We don't know.
I know. Yeah, we don't know.
We found we just find more and more functions in those sequences, and I'm parodying various creationist folks.
I know.
Like I'm anticipating a clip of Dr. Dan becoming a creationist and then playing that clip, that little round.
We're going to play that.
Look, one day if I sell out for more money because academia pays terrible, I would be a better young Earth creationist than literally every young Earth creationist doing it right now because they're all terrible at it.
You would, yeah.
I could do a better job too. So like Donnie Badinsky, standing in poop, he says that the onion has 16 billion genes, or 16 billion nucleotides in its genome. It's got to be that big because the onion may have to survive in the Arctic. He actually said that out loud somewhere. Basically, it has to survive. Nice. Yeah. So that's why God treated him that way. But then the other members of that same family have around 6 billion nucleotides because they didn't have to survive in all. I don't know what the fuck. Okay, moving on. Ready?
Hit it.
I have a creator can do anything. I don't know how else to repeat it. I will point out, I myself have seen in the lambda interferon 3/4 the same mutations in the pseudogenes of chimps and humans. That has been an argument for common descent. Mm-hmm. Yeah. But also it has said, okay, so this caused damage to both lineages. We have immune disorders in both lineages. Now, this could also be a miracle of God's judgment, which is consistent with the Bible. But, the thing is, you're straining at gnats. You have much bigger things to deal with nested hierarchies than straining at this gnat. And I get after creationists. I said, you're letting people like Dr. Dan define what the real argument is. The real argument should be, how do you have all these coordinated changes? If you're going to strain at those little gnats, you're letting camels through, and those are the nested hierarchies.
And so what the hell is he babbling about with this camel and gnat thing? Is that through the eye of a needle?
Yeah, it's some Bible reference, and he says it like 10 times in the stream.
Yeah, straining at gnats is a criticism Jesus made of the Pharisees about them being so picky and meticulous about certain rituals. And then the camel going through the eye of the needle is another reference, reference to something Jesus said in one of his explaining one of his parables.
So we're not doing the camels and we're doing the gnat. I don't know.
I just want to appreciate the fact that he's mad at me and other creationists because they're letting me define the terms of the argument, to which I say, damn straight I am. That's right.
Yeah. Nestle got it right here.
So many, I'll just say, since I've been making the nested hierarchy thing a thing for the last few months, professional creationists are rumbling about it, and they're all trying to talk about it and come up with explanations and stuff. And it makes me so happy to see them having to respond to stuff that some rando is putting out on the internet. Yeah, that's right. Professionals. You got to respond to me. Damn straight.
Yeah. I love it. Yeah. Pretty cool. We got a few more seconds here, and I think
he gets mad.
Oh. Anyway, okay, I got a few more seconds here, and I think he's going to do it again.
That have to co-evolve. I can't tell you, this is unbelievable. You're not even dealing with nuclear localization signals. You pretend it's not a problem. You don't deal with binding interactions. You don't deal with transcription factors having to be tuned to the DNA promoter regions when they evolve. You're straining at gnats, letting camels through, and a creator can do anything. And what Dr. Dan is doing is defining what a creator ought to do. That's not right, and that's fine. Look, I actually did credit Dr. Dan that he made a very good analysis, but I've always complained to him, you're straining at gnats, letting camels through. There are big elephants in the room. You pretend they don't even exist. You just deal with this little gnat. You pretend like you know.
Well, how are you going to get the elephant through there?
Pretending they don't exist.
So basically, he's trying to minimize Dan's argument rather than deal with it, rather than actually refute it.
But the way he's doing that is to say the creator could make the hierarchies look any way the creator wants, which fine, fair enough. But now you're proposing an ongoing miraculous intervention in mutations that happen. Which, great, we're not in science land anymore. So thank you.
The world is a comic book where anything can happen.
Right.
And why would you do it in pseudogenes?
Why would it happen in pseudogenes? Yeah. Why were somebody in my chat on my channel said, why did the chimps get the same curse as the humans?
It's like he got so close to putting the pieces together, but I think he caught himself and then started ranting.
Right. Yeah. Well, his complaint that we don't look at the interactions, that's all biology, that's all molecular biologists do. They do it all day long, every day. That's all they do is look at those co-evolving interactions, basically.
Anyway. So what's your next timestamp? Because I got a bit of I got a jump in mine of about 5 minutes.
Okay, mine is at 41. Let's see, 41:22.
Well, here, can you go to 37 first? Because there's this pencil analogy that I want everyone to see because it's just...
the pencil. Oh, my God.
I want everyone to see the pencil analogy because this is Grayson saying... Grayson said, look, in order for, God to intervene like that, it would basically be a deceptive creator. Right? And that's not, that's not allowed in most, theological interpretations. So
I miss that bookmark.
I will say, I think Grayson is really good at explaining this stuff.
Sorry, say that again, Andrew. I stepped on you.
Sorry, I was just saying, I really think Grayson is good at explaining all this stuff and interacting with creationists.
He did a great job.
He is.
Yeah. Grayson, if you see this, you did an awesome job in the stream.
Yep.
Yeah, and he caught a lot of the points that I was like when they started out, I was just screaming, like what I would say. And then he went ahead and said it. He did a good job. Yeah, that's pretty cool.
Great minds think alike.
Yeah, well, he's got a great mind. Somebody does. Anyway, okay.
Look like they're inheriting the same mutations from a common ancestor, which is what we see.
Rebekah, can I just share one little slide?
No, boy.
But is it going to stay on this topic, Sal? Because I don't want to go to another topic. Okay.
It is. Because this is addressing Grayson's point about God being deceptive. So if we look, this is just a metaphorical illustration here.
Then why show it? Why
you could add the little pencil, the bent pencil. I had a picture of a bent pencil. Okay, is God being deceptive because that pencil looks bent?
No, because God doesn't exist.
Is God being deceptive because that pencil looks bent to our eyes?
No, because the consensus in science is not that the pencil is bent. The consensus in science is that evolution happens.
It has nothing to do with consensus. It has to.
Okay, should I go on a little further, or?
No, I think that's perfect. I think Grayson nailed it right there, which is the whole point. Is no, this is not being deceptive. One, because just physics, so we know how physics works, but also because nobody thinks the pencil is actually bent. In phylogenetics, the consensus in the field is that the relationships are real, and if they're not, that means the deity that made the mutations happen to make them look real is being deceptive, right? Is actually deceiving people, right? This is so cringy. I don't know why. It's bad. That's all. I just wanted everyone to see that and appreciate it. I had to suffer through it. Now, this is my third time, so you all have to suffer through it at least once.
Well, I hope it's been therapeutic for you, Dan.
This is really the secondary purpose here is hanging out with y'all wonderful people and talking about science and creationism. But the primary purpose is that this is really group therapy because we've all had to experience this.
Right.
I just put in a shameless plug in chat for your people that subscribe to my channel because you got like 60 watching and I have like 9. So anyway.
I remember what I was going to say earlier. It's almost like the Omphalos argument, but for evolution. That it's all an illusion.
Yeah.
Yet they can't seem to demonstrate it, as Nestlig's saying.
Yeah, the real thing that focuses on is when it comes to these non-functional regions, right? I mean, it could be functional because they're a certain size, but the details of what's inside of an intron do not matter for the most part. There's only about 6. There's only about 6 nucleotides that matter in an intron, and that's so it can get spliced out. And the rest of it, very seldom has any function. But they'll say, "Oh no, the whole genome is functional." Well, then, if they believe that, then they can't believe in genetic entropy, and they can't believe in mutations at all. They can't believe in this bullshit they're saying that we're mutating out of existence, because that requires that the entire genome is not functional, basically. So anyway, let's see where we going next.
Jump to your thing.
Okay, that was 41. Okay, assuming the starting point.
The swear jar speed.
What's that?
I said, don't forget the swear jar.
Oh, did I swear? Did I swear?
Not badly. I don't think it was
bad.
I'll put $5 in it.
Just throwing that out there.
God damn, I hate when I swear. 4122. Okay. Let's see. Okay, 4122. I gotta keep scrolling down on this badly designed bookmark program. They could have done a better job.
That's the thing, Grayson, is that, so remember, I'm not asking for 15 bases out of 15 that are common. We're both knowing what the variation is now. The question is, how do we get back to a starting point? Right. And what I'm saying is, and I'll try to say this in a way that can be understood, but you're assuming the starting point for human, chimp, and gorilla. Yeah. And what I'm saying is you don't have a scientific basis for that starting point.
No, I'm not assuming any. Look, I'm saying based on combinatorics, how many possible starting points could you have for 3?
Okay, we should probably address this. There's this confusion over were you there? Okay, if you have chimps and humans today, we have 2 extant species, and we look at the genomes and we infer the commonality going back. We don't assume it to begin with. We start with what we have today. Now, I had a conversation with a really dumb guy on some channel who was saying, you can observe today, like you can observe my genome and my cousin's genome and tell that we're related, right? And he said, but you can't tell basically what you have no common ancestor back there. So he was saying that we couldn't learn anything about relationships because we don't have that common ancestor in the past. And I said, but you just told me that you can tell if you're related to your cousin, right? So, I mean, obviously we can infer common ancestry, and he didn't get it. He didn't get it at all.
Isn't that how they catch serial killers and rapists?
Yes.
They look at present day genetics and see who was the father or whatever, and they know who it points to. At least usually they get it down to like 5 or 6 people that could be the suspect, and then they take it from there.
Yeah, it's like if if you introduce your mom to somebody, you say, "This is my mom," and they say, "Well, how do you know? Were you there, or did you?" Well, I guess you were, but you didn't observe it, right?
The question that I hear a lot that I like to ask is just say, okay, you accept these basic things, and creationists, for example, love to cite pedigree mutation rates, and you say, okay, when does it stop? When does that pattern of inheritance stop working?
You'll never give an answer.
And you will never get an answer, because the answer is at the kind boundaries, wherever those are, but they can't articulate where that boundary is or how we can tell. And the whole thing with the nested hierarchy thing, that this argument that I'm trying to popularize here, is that if there were separately created kinds, we could see that phylogenetically, because the pattern would break down at those boundaries. And the fact that it doesn't means that there are no boundaries,
obvious.
right?
Okay, and then we're going to go on to
like in a single lineage when that doesn't prove they're not related to each other. Just a misunderstanding of how phylogenetics works.
Yep, and then we're going to go on to this histone bit. Do you want to cover that, or do you have? What is your next bookmark?
I don't even look. I have so many bookmarks, and most of them are like very repetitive. It's irreducible complexity stuff. It's Rebekah, but what was the starting state stuff? And I'm going to do a deep dive into that 2016 paper that she's talking about, the bomb paper that computationally looks at basically it does a number of tests of the strength of common ancestry and separate ancestry. And I'm going to do a deep dive into that. At some point, I would probably think in the next month or so is when that's going to happen, because it involves some fairly complicated computational methods that if you don't understand the methods, then like you're not going to be able to articulate how that paper works. And so I'm going through, and this involves like diving through the references in that paper to then see what the methods are in the references that they're saying, we did it like these guys did it in 1995, and then you got to go back. But it's a probabilistic method, which means there is no fixed starting point, is the short version. And I mean, just that right there, it's probabilistic, therefore there's no fixed starting point. Like in my model, there was a fixed starting point, right? When I did my little simulation thing, this is the starting sequence, and then we mutated. In that paper, that's not the case because of the way the model works. And like that simple little realization deals with like an hour's worth of Rebekah's problems with that paper.
She seems to be conflating the two.
My model was much simpler because the whole point was you could do it on screen in a stream.
Do you hear that, Rebekah? You better watch that video whenever it comes out.
It'll get here eventually. Not like I've work to do or anything.
The next one I got is Cell Gish Gallops on the histones, and this Behe paper from 1990. Did you see that part?
I have no idea what paper it is. Yeah.
I looked at it. So let's just listen to it for just a second.
I have some data points. I just realized this.
Data points.
The evolutionists are cherry picking certain things because this does not happen for histones. Histones are identical practically almost all the way to yeasts. So the histone in histone 1, histone 3, histone 4 in humans is identical to chimpanzees. So you're going to take the most variable proteins and start making an argument in nested hierarchies with there. See, this is cherry picking on your part. There's no variation in histones, and this is
full genome, Sal. Full genome. We're not just going to look at certain proteins.
Okay, so he mentions this Behe 1990 paper. Histone deletion mutants challenge the molecular clock hypothesis because this group of researchers deleted part of a histone gene in yeast, and the yeast basically survived. But their cell division was slower, and there were a few other things that didn't happen as well. And if this organism was stressed at all, certain things wouldn't work. And histones are basically highly conserved, right way down across all the eukaryotes, right? And there's a lot of reasons for that. One of them is, but one of the histones I think has like 50 different copies, basically that are all fairly highly conserved, and they all come into play at different points. And so basically, he's taking something that is extremely complicated in 1990, where they had nowhere near enough information to understand how histones work and how nucleosomes work. And he tries to make a point out of it. So Sell is reaching way back in 35 years ago to Behe's 1990 paper and research that was done in the 1980s. So I don't know if any of you guys can even remember the 1980s, but.
And the other thing here is that this is another example of like, they ignore this, they're cherry picking, they don't talk about this. Yeah. I literally talk about this exact thing when I cover phylogenetics in my 200 level intro to evolution class. It's the first specific class you take in evolutionary biology. If you're going to take evolution classes, you take the 200 level course. It's called Fundamentals of Evolution. And on one of the days when we're talking about molecular clocks and phylogenetic analysis and stuff, we talk about how it's really important to pick your regions of your genome carefully because they evolve at different rates. So if you have a region that is under very strong purifying selection, which is to say selection that keeps it the same, any variant is selected against, then those regions are only useful across very broad groups. Because if you were to try to compare humans and chimps and gorillas in these things, and it's funny that he brought up another one he mentions, I think, is cytochrome C oxidase, which is one of the examples I use in class, which is remarkably consistent because if it breaks, your cellular metabolism doesn't work and you die, right? So it has to stay the same or you have problems. Right. So you can't use that to compare across like humans and chimps and gorillas and orangutans, but you can use it to compare across prokaryotes and eukaryotes, right? Because everyone has it and they're different enough across those large scales. But you can use other parts of the genome that do evolve faster to compare across the narrower groups. You just have to be careful and conscientious of what genomic regions you pick. And Grayson makes the point here that it's not cherry picking if you just compare everything, right? If you just compare the whole genome, you're not cherry picking.
Yeah.
It's
consistent. And the point of, go ahead, Andrew.
Or it's consistent either way, no matter what part of the genome you're using.
Yeah.
For the most part.
The thing about the histones was, okay, the sequence is highly conserved, but they deleted it and the organism survived. But then if you look a little bit deeper, the organism didn't survive quite as well.
There's still costs. Yeah.
And there's a whole lot of other complications with histones. And Brandolini's law, Sell brings that up, shows that 1990 paper, and now you've got to read roughly 1,000 papers, up to the current time about nucleosomes to figure out why this happens. So anyway. So now I'm on to the humans and gorilla paper, the comparison. So there's a little snippet on that. What is your next bookmark, or what would do you have another one you want to address?
So there's this whole bunch for like 10 minutes of Rebekah with the human gorilla thing, and it all comes down to her not understanding that they didn't pick a fixed starting point in that paper. And it's probabilistically determined rather than specified, right? Specifying a starting point for each, lineage. So if you want to play any of it, go for it. But like, it just comes down to Rebekah not understanding the methods in the paper.
Is this the paper?
So if the next really, oh, sorry, Andrew, go ahead.
Or is this the Baumanol paper? Sorry, am I on a delay here?
Yeah.
Yeah.
The next really fun one I have was at 55 minutes because Sal starts to go a little bit ICR on us, if everyone knows what that means.
Oh.
Okay.
That part's fun.
Okay. 55. I have a bookmark much after
mental
that.
prediction to see if it's confirmed.
Yeah, the entire 10 minutes was on the human chimp genome comparisons. Okay, I'm at 54:34, so I can run it from there.
It could be a natural hotspot. We don't know a lot about how DNA may change. The evolutionists keep putting this random distribution, and I've already pointed out examples. You can't universalize that random distribution, especially like with histones, and you erroneously said, even after I cited the histone deletion mutants, that conservation is due to natural selection.
He's talking about interferon, I think, and he's basically postulating that there is a singular hotspot, like one nucleotide, right? Which kind of misunderstands hotspots, I think. Hotspots can be a little more general than that.
Hotspots are regions, not specific sites.
Yeah, and they can pull out a hotspot talking point anytime they want. So that's as bad as pulling out "God did it." So anyway,
I want to insert a quick aside and kind of summing up principles to glean from this so far. Sal has taught me that number one of the faults of creationism is they often rely on very old papers. And another fault of creationism is they often have people who are not experienced enough in the field they're talking about to know that there have been papers and discoveries and that clarify, explain, or even overturn a damn paper from 1990. Here's my $1 for the swear jar.
Yeah. If it only costs you $1 to say damn, but if you drop the F-bomb, it's $5. So anyway, I don't know where I was going with that. Anyway, okay, I'm gonna go play it.
Natural selection. So the other thing is, as I just looked at this up, statistical evidence for common ancestry, they totally ignored the point that I was saying. The nested hierarchies that co-evolve will not work if you're actually looking at the binding interactions and things like nuclear localization and transport processes.
Oh,
evolutionary biology totally ignores that, and Dr. Dan
totally
knows that is he suspects.
My prediction. Hold on, Dan. Is no, they don't. Because this little guy waving his arms and high pitched voice, he is not an expert in the field. Therefore, my prediction is he is not knowledgeable enough about what's been written, what's been done, and he's probably relying on old papers.
Yeah, and I've got five papers that I downloaded in about 5 minutes. New papers on the nuclear localization signal and how many different kinds there are and how many how many variations there are to that little lock and key, that sort of thing.
Yeah.
Anyway.
All you have to do is Google it to see that he's wrong. Like, you just Google it and see how many studies there are looking into this exact question. They ignore it. No, we don't. Like, just Google it. You'll see how it's not ignored.
Google Scholar exists.
Yeah, Google Scholar is wonderful. Well, if you want to find out how did the liver evolve, just put that into Google Scholar and start reading, and you'll be reading for the next 6 months. Anyway, I've done it.
Here. You just got to read it.
Yeah, you got to read, and it ain't easy to read. Okay.
Yeah.
Going on.
It's going to come to bite you guys up in the next 10 years as we do more sequencing, as we do more protein testing, as we do more biophysics and biochemistry. That is something the evolutionists, all they do are these phylogenetic trees. They don't analyze the biochemistry, which is so important for these things to function.
I can tell that you really, really want to move on from the subject that we're currently talking about, so you can talk about your thing, Sal. I'm happy to do that.
That is the thing that is on your...
Grayson has a response here. I'm hoping I catch it to that particular thing, which is kind of cool. To be fair, Sal, you were saying that 10 or 20 years ago.
I'll just say one of the first things I worked on in graduate school was protein stability and how what they're called host range mutations in viruses. So mutations to their proteins that allow them to infect a new host. And basically how protein stability of those capsid proteins constrained what host range mutations could occur. Like that, literally my first semester of grad school, that was something I worked on. And here's Sal yelling about how no one considers that stuff.
Right. Yep.
Yeah, it's not like biochemistry isn't important.
Sal's getting a little excited here. He's starting to point. He wasn't extremely happy during this entire. There's one point where he pulsed like a teenager for about 5 minutes, which I thought was really wonderful. Anyway, okay. I think
when you can tell he's losing an argument.
Have we gotten to where we want to yet, Dan, or should we keep going here?
Oh yeah, I think jump to wherever you want to go. Yeah.
Oh, let's see. Where do I want to go next? Well, okay, so at 59:16, he talks again about the 10-20 years. So this is where Grayson, I think, comes back, and I thought it was just wonderful. It's kind of the creationist narrative that with what we're learning today, it's obvious that God did it. And actually, it's just the opposite of that. And it's because they're just not somehow keeping current with the literature. I don't know.
You can find creationists saying that, oh, with all this new stuff in 10 years, it's going to be a revolution. You can find them saying that stuff going back to like 1996. Like in the modern creation.
Before. Yeah.
You can find it from before, but like in the modern creationist movement, you can find it from the 90s. Oh, 10 years is going to be a revolution. And nothing has changed in 30 years.
I mean, like we said, that's where most of their knowledge comes from anyway.
Yeah. Okay.
You guys totally ignore what's going to undo all of this. I can understand why you're going to ignore it. You're going to focus on the gnats.
I'm really not ignoring it now.
Because this falsifies it. And you're going to lose this in 10 years. In 10, 20 years, you'll be gone. Just like this whole thing about...
Sal, to be fair, you were saying that 20 years ago. But I'm not...
Boom!
There you go. Boom. True. Sal was literally saying that exact thing 20 years ago.
Yeah.
And I know that because I was there for it. Like, I have been interacting with Sal for that long. And he was writing exactly the same stuff 20 years ago. Yeah.
I think we need to put together a GoFundMe or create a foundation for people who had to go back and read Sal Cordova's stuff 20 years ago.
I remember. Here's some deep lore for you. If you want to Google it, it's really fun. Go look up the falling out between Sal and the Uncommon Descent blog, because that was a whole thing in the ID movement. Yeah. That was a whole thing back in the day.
I don't think I've ever done that. We should do that sometime.
Oh yeah, it's the deep lore.
It's great that we have someone in the community who's been around for so much of this.
It's not quite as good as the AIG CMI split because that involved accusations of like witchcraft and necrophilia. But you know, the mid 2000s post Dover ID movement crack up was pretty good.
Yeah. So let's see here. I've got now Grayson goes into this insect thing, insect metamorphosis. I don't know, should we skip all that or
I have all that. I don't know anything about insect stuff. Like not my field at all. So I cannot comment on most of it. But the point he makes, I'll just really quickly summarize the point Grayson makes, which is really cool, which is that the genetic similarities, the pattern of similarities we see in the living insects across insect biodiversity, that pattern of genetic similarity is correlated with the pattern of morphological similarity in the fossil record. And you don't expect that correlation unless they share a common ancestor. And that's actually one of the tests in the 2016 paper that Rebekah's been talking about, the Baum paper from 2016. They do looking at how these different metrics correlate with each other. And basically, the higher the level of correlation, the more that's in line with a common ancestry hypothesis rather than a separate ancestry. So this is, I encourage people to go and watch this because it is really cool. And Grayson does a really good job explaining it. But basically, it's about how the genetics of the modern species, that pattern correlates with the emergence of different variants in the fossil record. It's very cool. And it's very strong evidence of common ancestry.
Yeah, I'm signed down with the wrong account, so my stuff is not transferring. So let me go back and do that. Let's see, I got to change some stuff.
I think there are late 90s have tested that as well. I might have.
I posted the paper. I posted the paper name under my name, Mike Brusnak, but I'll do it again here under Answers in Atheism. And I think now my comments aren't showing up in both streams, so I have to put them in both. So it's Truman, 2019, The Evolution of Insect Metamorphosis. And there's just a whole lot of misunderstanding going on here. Rebekah's confused by basically by the thickness of the lines at the bottom and some things like that. So I'm not entirely sure. But it's like, no, I'm still
She
not
does
working,
concede that there is some strong evidence here, which is nice.
Oh, that's
yeah. Yeah, she does.
What's up, RJ?
That's kind of
okay. And then we let's see, we have cells data point, cano revival, and identification of bacterial spores in amber. And I went and looked at that one, and that's just bullshit. Yeah.
I looked for it and I couldn't find it. I have no idea what he's talking about.
I found it. It's cano 1995.
I found the salt one from he got the years wrong though, because there was the amber spores that was like 25 million years, and then there was the salt pocket one in New Mexico that was like 200 million years, but he said it was amber was 200 million years. I couldn't find that. I think he's either conflating the two or just making it up.
Is this
okay?
the
This
claim
one is
there have been living bacteria for 200 million years?
Yeah, that one.
Okay.
Which it was actually bacterial spores, and it wasn't in amber. It was in like a salt flat situation. Yeah. And
big difference between bacterial cells and bacterial endospores.
Spores, right? Yeah, they're different things. Yeah.
Yeah, it was in 1995 that they did this, and it was cano, and I posted the name of the paper, Revival and Identification of Bacterial Spores. And his point was, like, how do you know the bacteria look the same? I don't know. I mean, if you look at a bacteria,
right? Because they didn't have the cells; they had the spores, right? They didn't have the actual cells from however many million years ago. They had the spores.
Yeah. And I think they revived them, and they took a look, and there was conservation, but I don't know if they found an exact same copy.
Grayson makes the point that there's conservation in horseshoe crabs. Yeah, they look the same from the ones you find hundreds of millions of years ago in the fossil record. That doesn't mean they're genetically the same.
But shit, if they were able to revive those spores, then they could get the genome too, right?
They should have, but it was 1995, so like,
right? So they didn't, and they apparently haven't.
Like I said, I looked for the sequence comparisons and couldn't find it. I found the amber one that the years were wrong from what Sal said. I found another one where the preservation method was wrong from what Sal said. I couldn't find the exact thing that Sal described, and in none of these examples could I find the genome sequences. So take what you will.
Can do a follow-up study and put that to bed.
It would be cool. That would be a really cool study to do.
Yeah, that would be cool to get a genome from an early bacteria, do comparisons. Okay, so now we go to neutral theory for just a minute, and let's see. Then we got Tiktaalik. We got poor little guy. Peace can fly joins.
We got to do poor little guy.
We'll do poor little guy. So how much time we all got? So who's burning out?
Let's do, I think if we do poor little guy, and then there's a little bit, the last note that I have is 134-ish, 134.30, give or take.
Dr. Dan concedes?
And yeah, exactly. So if you do poor little guy and Dr. Dan concedes, like that puts a nice bow on everything because it I think, clearly encapsulates where Rebekah and Sal are coming from.
Yeah. 128, 120. Let's see, where are we here? Okay, so
like 127-ish, 127 something.
It's 128, 129.
Yeah.
28, 129, 126. Oh man,
they phrase it. You're conceding the point. You're admitting XYZ.
128. Okay, so it's around right here, I think. Let's see if we can find it.
And we don't see them transitioning to other forms, except in very rare cases where you can say something like a Tiktaalik looks like it might be a combination of things, but that's very rare. But we've got to get from the first living organism to the diversity of life today. But everywhere we look, it's like boom, wings, boom, eyes, boom, all these, fully formed systems. Well, how did you get to the eye? Why can't we look and see, poor little guy, he almost had an eye, but he didn't.
So she doesn't know about eye evolution.
Why do we see fully formed eyes? Why do we see?
Okay, sorry.
Oh, poor little guy, he tried to evolve himself a wing, but look at it. It just wasn't flight worthy. It was just like a little nub.
Because that's not how evolution works. You don't just
that was wonderful.
That was the moment right there. That was the moment right there where I'm just like, and I don't know if Rebekah, if you're going to see this, sorry, but that was the moment right there where I was just like, I'm not really going to listen to Rebekah anymore.
No,
like, there's either no interest in learning anything about this, or genuinely just doesn't know and doesn't care to know anything about, or she knows better and she's just lying about how evolution works, right? There's no good option here, because these conversations with Rebekah have been happening for what, I don't know, like 4 years, as far as I don't know. It's been a while, right? I know, she's been, I've talked to her a bunch. I know she's gone on with like Jackson Wheat and Dapper a bunch of times. Like, people have explained the basics of how evolution works. And either she refuses to assimilate any of that information into her framework of how the world works, or she has assimilated that information and she's just misrepresenting it. And I don't know which one of those is the worst option, but like, it's one of those two things. And I guess we're just continuing the trend of Dan is disappointed by creationists. Oh, well.
Yep. There's a little piece here I just have to show.
With a different body plan. So how does that happen? Where in the world do we get that? Because the caterpillar can't reproduce. So if it starts out as a caterpillar, it doesn't have millions of years.
Anyway, this is just real confusion over metamorphosis and the development of metamorphosis, and the stages of insect development, basically. And when it went to metamorphosis, they had the same stages, but they happened at later times. They were kicked off at later times, and she has a real limited understanding of how that could have evolved and how that did evolve. So anyway,
it's a good resource for this is actually I think it's in Your Inner Fish, but it might be in Neil Shubin's other book, Some Assembly Required, but it's in one of them. I think it's in Your Inner Fish.
One of the Neil Shubin? Okay.
Where he talks about chordate evolution and how basically, like, you have tunicates that change their morphology once they kind of attach to a surface, but then all the rest of the chordates, starting with the lancelets, they basically stop their development early and they stay in the more the kind of the earlier stage of development, and they stay free living throughout, and they can swim around and they have the notochord and everything throughout their lives. Now, lancelets actually end up burrowing anyway, but that's besides the point. But then with insects, it's a similar thing where instead of stopping their development early, they're basically just expanding the time range for embryonic development because they need to acquire the energy. So they need to have a free living stage to acquire the energy to then complete embryonic development. So the larval stage is kind of an intermediate free living embryonic stage, where in most other organisms and in the non-metamorphosizing organisms, that's all done embryonically, right? But then in the ones that do metamorphosis, they become free living at an earlier stage in development, gather the necessary calories to complete development, and then metamorphosis is essentially completing that process. So I don't know. I don't know what the problem is.
So if I took every question that Rebekah asked just now in her little rant and put it in Google or ChatGPT or even maybe some website where you can ask a scientist something, I think the answer would be pretty forthcoming, don't you? I mean, if you just take the time to do the research on those questions.
And let's just be clear, to do this, you need to regulate the timing of development, but we know a lot of the genes and mechanisms that regulate the timing of development.
Right.
And humans are interesting because we kind of develop a little bit outside of the womb too. We have this delayed development, basically,
right? Because our heads get so big.
Our brains. Yeah.
Exactly. So we kind of do that same thing where for the first, they call it, the thing you hear this term, the fourth trimester for humans, because those first few months of human life after you're born, that stage is basically done in utero for all other mammals, basically.
Yeah.
And the reason for that is our brains get too big. We wouldn't be able to make it through the birth canal after those 3-ish additional months pass, so that human babies are born way less developed than a chimp baby or a gorilla baby or a squirrel baby, right? And it's because of our big brains.
Yeah. And one of the things that differentiate humans from a lot of other animals, not whales, but a lot of other animals, is that we have many more neurons. Basically, there were a couple of modifications that when the neurons were starting to divide and create the number of neurons, that we ended up with a tremendous number of neurons. And because of that, when a baby is born, the baby has to cull a lot of connections that have formed. And so there's a lot of development that goes on in a child in that first year or two. Basically, that happens a lot faster in a lot of animals. So, and that's sort of a neurology perspective. But now we're going to get to Dr.
Creationists say chimp and human brains are pretty much the same. They're just ours grow for a longer period of time, so they grow larger.
Yeah, they produce more neurons, quite a few billion more. But then whales and I think dolphins or porpoises have a hell of a lot too. And whales have more than us, which is really interesting. Yeah. You wonder what they're doing out there with them. So
heterochronies.
I bet they're doing a lot of interesting topic. Yeah, I bet they're doing a lot of remembering. They've got a lot of episodic memory going on. Anyway, okay, we're at Dr. Dan Concedes. I want to see how. I forgot what you could.
Is it? This is the part where my career ends. So.
Okay. Good. Oh, no. Good. And you're going to go to work for ICR now. Okay. At a higher, double the salary. Double the salary.
Hey, if they'll have me, if they'll pay me, I'll do it. Yeah. I'll do it in a heartbeat. You want?
I would too. I'm ready. Okay.
I'd like to add one thing.
Sure. And then let's let a beautiful talk after that. Yeah.
A lot of evolutionists love the fossil record because it erases all the important details. You have to deal with stuff at the molecular level to argue these changes. And when you have large protein, you have major new protein families that are needed to make these changes, which we haven't even sequenced. We don't know. We haven't even characterized it. But once you have to start to account for that, the problem starts to become really difficult. That's why I'm really glad Dr. Dan conceded. All major protein families do not have a common ancestor. So to the extent that you might have to have a caterpillar that can now become a butterfly, we don't even have an accounting of all the new proteins that have to be there, that just have to sort of pop into existence. Just looking at the fossil record and saying that evolved.
Okay. So this is Protein Orchard again. So this is protein, and he actually, what did he say? We haven't even looked at these protein families.
Haven't even looked at it, Mike. Haven't even looked.
We've looked at them all. It's like
we don't know what proteins you need for metamorphosis. Right. We haven't even looked at it.
Oh yeah, and the thing is that the amount of proteins, the amount of changes in the genome, in order to create a very large morphological change, is surprisingly little when you really start to look at it. When you start to look at gene regulatory networks, how a limb or a fin turns into a limb doesn't take a hell of a lot of modification, actually. So.
Yeah, because you have these template proteins that basically say extend, branch, fold inwards, and you're just changing the pattern of those expressions. Yeah. Whereas, like, so if you look, for example, if you look at an arm, right? You look at an arm, it's a combination of the extend and branch genes being expressed, right? So you have one bone, and then you have two bones, and then you have a bunch of bones, and you end up with 5 digits, right? It's so you're doing the extension and the branching, and the pattern of those genes gets you your different arm morphology. So, like, for example, bats who have their wings, and bats, it's their fingers, right? Form the framework for the wing, whereas birds have lost the fingers. Bat wings are mostly their digits with the membrane in between. And that just means that the extension gene, and I think it's a Hox gene that does this. I could be wrong, but I think it's a Hox gene. I'm going to give myself 70% confident in that. And that gene is basically expressed for a longer period of time in bats after that final branching pattern is expressed. So for us, our fingers are a very small fraction of the length of the rest of our limb. But for bats, the fingers are actually the longest part because the extend gene stays on for much longer, right? And that's not a new structure and it's not a new protein. It's just changing the pattern of expression of existing genes.
Also, you mentioned
the
between the fingers in humans, for example, obviously the membranes between our digits as we're growing undergoes apoptosis.
It goes
Same
away.
thing in bats isn't.
Right. We have the teeny little ones. The
Right.
bats keep them. Yeah. Mm-hmm. Which is
Same
so you're
thing.
shutting off a signal rather than building
Duck's
something
feet,
new.
too.
Yeah.
Yeah. The autosomes, basically the fingers, are kind of a later development from fins. They used to think that the individual rays on the fins turned into fingers. It was a little more complicated than that. But anyway, there's the stellopod, pseudopod,
goes over
and
that too in his
autopod.
book.
Yeah. It's really interesting how the thing morphed and developed. But it's all about spatial timing and two dimensions, spatial and time dimension, basically, as to how these proteins are released, and it will just drastically modify a limb from one animal to another. And but we have that same pattern, and it's amazing to look back at fish.
Anyone who's interested should look up the term heterochrony and heterotopy if they want to learn more.
What was the first word?
Sorry, heterochrony and heterotopy, I believe.
Oh, okay, heterotopy. Heterotropy? Yeah.
Heterotopy.
Yeah.
Time
and space, basically.
It's been a while since I've looked
time
at
and form
development.
Yeah. Yep.
So some of you may know that Sal recently did an online talk at the Evolution Conference, the 2025 Evolution Conference, which you can find that online, and you can find me and Zach Hancock talking about it on Zach's channel. And I bring this up because this thing with the protein orchards, I actually said, Sal, you should talk about this. He ended up not. He talked about something else. But
right.
I have said since then that Sal should try to give a talk on the protein orchards thing at an evolution conference in the future and see how that goes. Because I've explained this to him a bunch of times that he's like spiking the football on this thing where it's something that every evolutionary biologist agrees on. He's like, they concede there are different protein families. Yeah, because de novo proteins evolve. That's one of the things that happens in evolution. And you should go to a conference, Sal, and talk about how, hey, look, all you evolutionists agree that proteins don't share a common ancestor, which is to say de novo proteins have appeared. And he should explain at a conference why this is a problem for evolution and see how that goes. I think he should do that.
Yeah.
You know, this reminds me of Mike. Here you have Sal, who is either himself ignorant of what's been going on in the field for 20 years, or he just wants to pretend that nothing's been going on in order to bolster his clickbait base or whatever. We have Mike, who's made these really interesting observations about genetics and, the comparison of the chimp and the human genome. And he is really concerned that doesn't know something, that there's something out there he doesn't know, that he hasn't read, and he wants to find it so he can, include it in his thoughts and his presentation. It's a big contrast, shows the difference between science and these creationist folks. They want
they basically give
superficial appearance of scientific knowledge rather than actually pursuing scientific knowledge.
Yeah, and I never have certainty. I'm always like even now that I found something in the genome that's kind of remarkable, I'm still not sure if the genome is right. And I'm still not sure if did I make a mistake? And I double checked it about. Anyway, Dr. Dan and I are going to talk on August 13th, I think, about chromosome 2 and some of the stuff that I've observed in the latest genome. So anyway,
I'm really excited for that. It's really cool stuff, folks.
I hope I can put it together in a, I'm not good at this stuff. I'm good at doing the research. I'm not good at presentation. Maybe, hopefully I'll do okay and we'll be able to get it across. So Pigskin Fly comes on and he talks about editase, which is a protein that repairs some of the genome. And the way he talks about it, he thinks that this protein has to understand the entire genome in order to make repairs. That it has to have like a little book copy. Yeah, he is so confused.
I think you're giving Pigskin Fly way more credit. He just came on to do his debate bro shtick. He was not interested in having a conversation. He was constantly, and he was constantly going off topic to like, I want to ask you this very specific,
right,
setup. He was just a complete bad faith actor the whole time, and I can't believe I sat through the whole thing. It was obnoxious.
Yeah,
Pigskin Fly, you're obnoxious. Life.
I've had conversations with him. He doesn't understand how GPS works.
Yeah. So it was Immutable or else Grace, and I can't remember which, that we're talking about a crime scene investigation. And Pigskin Fly said, "Do you think crimes happen by natural causes?" It's like, yeah, I do. I do. I mean, what do you think? We kill people supernaturally? I mean,
right. It's like, there was a material cause to the murder. Yeah, that's
supernatural cause. Yeah, it's like, God, I want to use that as my defense. I want him as my defense attorney if I ever get in trouble.
I'd like to raise another question. Earlier, when Sal was doing his God can do anything or God can change anything that he wants to, God can do anything. Right. Why does Sal, and I think Rebekah, although I'm not sure, continue to use the word natural? There is just no such thing as natural.
Yeah.
If God is bing, 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 just making all these changes throughout history and time.
I have that exact thing in my notes. So in the part where Rebekah was talking about separate ancestry, there was the line from that paper, separate ancestry by natural processes. And what it means in the paper is that once you have your individual lineages, the evolution that happens subsequently is through the genetic processes that we understand, right? Mutation and selection and whatever, right? Recombination, right? And the context there is just no divine intervention, right? That's what it means. And that was completely missed. And we didn't talk about that part specifically, but I have, Scott, it's so funny you said that. I have that exact thing in my notes. Yeah.
There's something really
There's
a really interesting tension here that is all throughout creationism from when it started back in the 1950s, that they want to scientifically prove that evolution can't happen. They don't want, like for the flood, right? They will not invoke miracles to create the Grand Canyon, right? They have to use science to do it. So they're stuck in this weird world where at any time they can bring out miracles, but they don't, because they want science to tell them that God exists. So they're doing.
It's always been for political purposes to get into the classroom.
Oh so that too.
It started.
There you go.
In the 70s.
It's just Christian nationalism.
Yep.
They have made some strides in the last couple of years, I'll tell you.
They sure have.
Pretty amazing.
Yeah.
They're banning books. Yeah. And they're doing a whole bunch of crazy stuff. Anyway, let's see what else we got. Yeah, that was it. Pretty much covered that. Covered the hell out of that. Anyway. So,
my just discovered that Sal and Rebekah are collabing again.
Oh, she just found out that they made up over their.
I guess so.
Yeah.
Like I said, my Sal either got less homophobic and Rebekah got, or Rebekah got more homophobic, or some combination thereof.
Yeah.
It's just apparently all that talk about being somewhat positive towards the gay community isn't so bad anymore for Sal.
Yeah, and the interesting thing is that this was not Rebekah's fault. She did not decide to cause it. It was Sal who decided that he could not live with her opinion, basically. Yeah. So he kind of stomped off, right? And she was a little shocked by that, I think. So. Anyway, because hell, we can disagree, right? I don't know. But Cell's little Wooly gets confused.
A frequent occurrence from now, or will this be a one-off, do you guys think?
I think it's going to be frequent. I think it's going to go on for a while.
Time will tell.
Yeah,
because they mesh so well together. I mean, their misunderstandings are so intermeshed that they do really good. So, anyway, okay, guys. Anybody have anything they want to say or mention? What's happening? What's up?
Well, I will say that next week, in 2 days, as of this live show right now, which is on Monday the 7th, in 2 days, I am re-airing my conversation with Rebekah, where I walked through the common ancestry versus common design model on her channel. So if you just want more of this content, come check that out on Wednesday. And then I have a few things down the road that are coming up the week after that. I'm going to do another round of that the Creation Conference. I've got a Randy Galuza talk coming up, and I'm lining up some really fun guests for that one. And I finally am almost ready to record a video I've been working on. It feels like forever. It's been like a couple of months of creation myth video on the post-flood hyper evolution thing that creationists require. And we'll talk about how those numbers just don't work, and it's really fun.
Okay.
I'll have that video out eventually, I promise.
Let me see here.
That kind of meshes with your nested hierarchy demonstration too, doesn't it?
It kind of does. Yeah, it's nice how it all kind of goes together.
Yeah. Yeah, and if I ever write that Python program I was telling you about earlier, I will get your input on that.
Absolutely. That would be cool.
Yeah.
I did a little in grad school, but like just this much. And yeah, it's very cool.
Yeah, and what do you use for a tool to find basically find the trees?
What do I use? So I haven't done this for real in a long time, but in my lab in grad school, we used a program called Beast, which is a Bayesian phylogenetics program, and we had at the time. We had at the time you would consider it we called it a supercomputer. Nowadays it's just a really powerful desktop computer, but and we called it the Beast because mostly what the Beast did was Beast runs, and it would you'd set it up and let it run for 4 to 6 weeks to do your iterative thing. It would just go.
Oh my God.
Yeah. And it had, we had
the uninterruptible.
It was, what was it?
Does it now in about 30 seconds, I think.
Oh, I'm sure they do it way faster now. Yeah, because I'm talking, I was in grad school 10 years ago. I graduated 10 years ago. So yeah, I mean, nowadays it's going to be
amazing how much computers have advanced with phylogenetics.
Yep.
Too.
Yeah, I've got there's a site that I use for the stuff that Demeter and I did, where you can send any RNA strand out to the site, and it'll show you how it folds. Basically, it'll fold the RNA for you, which is really awesome. And I want to try. I've got a really big RNA for some project I'm doing that I want to run through it, but I'm not sure how it's going to do on big ones. We've done it on small segments, like with 30 nucleotides or whatever. But anyway, all that fun stuff coming up. And let's see what else. Oh, yeah, maybe I'll get my shit together and do a thing on consensus sequences, because this is really something that we have to understand, is that these lock and key things that they talk about with proteins fitting, enzymes and all that, is not what you think it is. It's not like a lock and key. The only similar thing, and that's not even similar, would be a skeleton key, where one key can turn a lot of different locks. And there's these sequences basically that let me just show one just as a teaser. Let's see, share screen, and we're looking at window, and we're looking at that.
Reason why arguments from analogies don't
my God. Oh, there it is. Okay. So anyway, this AUG thing you see here, which is really high, where the AUGs are really high, that AUG has to be there. Now over here, it's mostly AG, and sometimes it's CT, and these other ones are not conserved at all. They could be anything. So when you look at these sequences, and there's a whole bunch of little ways to write sequences. Like if you look at the zinc fingers, they're extremely mushy as to what has to be there and what isn't there. And this is, let's see, this I forget what initiation codon. I forget what this is. But anyway,
AUG is the start codon. Yeah.
Oh yeah. Okay. That's what they're doing. So it's has to, has to be there, right? Has to be there. Yeah, and that's that chromosome 2 stuff. There's some interesting things with start codons and the problems with that. Anyway, and pseudogenes. Yep. Okay. So I'm done. I'm gonna stop my screen.
Everybody.
So
if you're watching this on my channel, subscribe to Mike. It's linked in the description.
Yeah, because I'm really old and I'm going to die soon. I'll be so disappointed if I don't get to 1,000 subs.
And as the designated observer of the live chats, I just want to say what high quality live chats we had tonight.
We did.
Yeah.
I was just really impressed. I love side chats of this quality.
Yep.
Thank you everyone for joining.
Thank you all for joining. And seriously, subscribe to Mike. He needs like 200 more people. Let's do it, folks.
Get him to 1,000.
And thank before I die, and thank the three of you. So I think it was Standing for Truth the first time I used that sales pitch about 4 years ago. I said, please subscribe to me before I die. He says, that's your sales pitch? It's like, yeah, I'm shameless.
There you go.
Okay, guys, we're out of here. So goodbye, Crew World. Yep.
Bye-bye. And thank you, Greg Helton.